Okay, so hello everybody. So I'm pleased to introduce the 5th edition of this workshop dedicated to JPEG DNA. Thank you everybody for attending this workshop. So today we have two excellent speakers who will talk about DNA synthesis for digital data storage from a chemist's point of view and from an information theory point of view. The two presenters are Anthony Jenos from LIMS laboratory in Japan, CNRS in Japan, and Andreas Lenz from the University of Technology in Munich. So each of them will give a presentation of about 30 minutes. And then, contrary to what we did that for the previous workshop, we will ask the question after each presentation, because Anthony is in Japan, so it's midnight for him, so it will be very late if we wait the end of the session to ask questions. So we will ask questions just after each presentation. So first of all, I will give the floor to Anthony Jenos. Anthony was formally trained in math and physics in France before receiving a PhD in DNA nanotechnology from the University of Oxford in 2011. He then moved to the joint lab LIMS between the CNRS and University of Tokyo to combine enzymatic DNA nanotechnology and droplet microfluidics. After a brief stay in Toulouse in CNRS LAS, he came back to LIMS in 2014 to conduct research at the interface of DNA computing and microfluidics. So, Anthony, the floor is yours for 30 minutes.
Okay, thank you. Is it working? Can you see my screen?
Yeah.
Good. Okay, so thank you. Thanks. Okay. Do you see my screen?
Yeah, but only part of your screen. Yeah, so if you can start.
Yeah, it just disappeared. I was just working. So I'm trying again. So let me check that. Yeah, we do the same manipulation that I did before. Okay, so this, is it working?
Yes, that's fine.
Okay, good. Okay, so thank you, Mark, for the introduction. So my name is Anthony Jenos. I'm a CNRS researcher, but actually I work in Japan in LIMS, which is an international lab between the CNRS and the University of Tokyo. It's a lab that was established 25 years ago. And today, so we talk about DNA synthesis for DNA storage, and with a perspective for information theory. So I can guarantee it will be 100% chemical free. I won't go into too much into chemistry detail, but more give you an idea of what we can do, what we cannot do, and what we hope we'll be able to do in the future. So what do I do for a living? So actually, I've been programming DNA for 15 years. I did my PhD, studied my PhD 15 years ago. And what I do is I use DNA actually as a programming material to assemble some device and system at the nanoscale. So for example, I work on making on DNA logic circuits. And the idea is that you can program reaction between DNA strands to actually compute stuff. Like for example, here we implemented a small logic circuit. We also use enzymes, and for example, we put some enzymes, DNA in droplets, and by programming the reaction, we could have some oscillations. So we can program DNA oscillators. We also make, for example, DNA gels, like by programming connection between strands, we can have a gel that has like interesting physical properties that could be useful, for example, for tissue engineering or drug delivery. And we're also working on making material with DNA. So for example, one of the highly investigated area is a DNA nanoparticle, where you can attach DNA on nanoparticle of gold, and you can make some crystals like this, which are expected to have interesting properties. And so basically, I've been using DNA for 15 years now. I've been ordering like thousands and thousands and thousands of strands. So to be honest, I have never really chemically synthesized myself DNA. So I've done a lot of enzymatic synthesis like this. But you know, now DNA chemical synthesis is so mature that basically everybody just buys it on the internet. That's the way, and I will explain you how it works. And it's something that's actually very powerful. It's just driving much of biology now, the fact that you can have a cheap DNA on the internet, just ordering from companies. And I will show you a bit how it works. So just to go back in memory lane, so the structure of DNA was reported more than 50 years ago, famously by Watson and Crick in a paper in Nature. So the paper was just very short, like just 2 pages. It was actually not peer-reviewed, and actually it didn't have any data in it, but it was basically the birth of modern biology. So very famously, basically, they based their model on the work of Rosalind Franklin, which was the first to one of the first to crystallize DNA, which helped to understand the structures. And so this was basically the beginning of molecular biology, and people understood that pretty much everything that happens in cell is because of DNA. And then in a coming year after, people just cracked the genetic code, and they understood the basic of the process. So this is how it goes. So you have DNA in your cell, which is double-stranded. So you have here, for example, you just imagine you have a sequence for a gene. And then this DNA will be transcribed into RNA, which is a very close molecules. It's single-stranded, whereas DNA is double-stranded. And then you will have a special molecule that will come and transform this into a protein. And then the protein, they are the stuff that are actually doing things in your body. And they will do this according to the genetic code. So the way it works is you just pack the letter by 3. And then, for example, AUG will give you this with this amino acid, and AUC will give you another one, and so on. And this is the basic of this is the base of the genetic code for all living organisms. And it just took a few years to crack this code. And if you if you're doing coding, then basically maybe you just notice something very very evident is that actually if you just make a single mutation here, if you just remove the A, for example, then you will shift all the code. So instead of reading AUG, it will start, for example, by reading UGA, and all the code will be shifted. And that's a big problem, because instead of having this protein, you will have something that's completely different just because you deleted one letter. And that's why actually it's very important for people doing biology to have a very exact sequence. Because if you just make one mistake in the sequence, then your gene is completely wrong. And that was actually the big driver behind the DNA synthesis, is how can we make DNA that is highly accurate. But actually it's different from the requirement of DNA data storage for the reason I will explain. And that's why actually there's a lot of room to improve the process for DNA data storage. So just to come back on this story of the central dogma, so people understood that if you wanted to control protein, you had to be able to write DNA. And the question was for a long time, like, how can you write biological code? So if you remember the sequence of the structure, of the double helical DNA was reported in 1953, but it took almost like 20 years, 30 years to be able to actually freely write DNA code. And for a long time, and it's still used a lot, the main method was just cut and paste. So it was, if the only way for you to write a program would be to cut some part from other program and then stitch them together. And to give you an example, for example, if you have a gene from a human cell that you would like to express in other cell, this is how it would go. You would do cloning. So you take the human cell, you would do some biological manipulation, and then you would find a way to cut the part of DNA that's interesting for you. You cut it off from the cell, and then you have just in your tube, you just have a small fragment of the DNA. Then you will use enzyme to insert this fragment of interest into this blue long circular DNA, which is called a plasmid. And this plasmid, actually, it's something that's meant to be expressed and replicated in bacteria. So you just put this circular DNA in a bacteria. The bacteria will just treat it like its own DNA. And very interestingly, it will also express the protein that you have put in it. So it's actually, you just do a cut and paste. Literally, it's really a cut and paste. And if everything goes well, then basically the bacteria will start expressing your gene. And this is, for example, the basis for expressing, for example, insulin or some biological product of therapeutic relevance. And it's still actually used a lot still by the biological community. So now what you have, literally you have Plasmid Repository. So just imagine it's like a software repository, but you have like Plasmid Repository. And for example, Agene is one of the biggest in the world. It's located in Boston. And they have a huge repository of about 100,000 plasmid. So typically the way it works is when you publish a paper, you're supposed to send the plasmid that help you to do this paper to this repository. And Agene will take care of that. They will replicate the plasmid. And then when other researchers need this code, they will ask Agene to send the plasmid, and they will use it. And it's actually very popular. It's still used a lot. And for example, typically, this is the way it works. I took an example of a plasmid that will code for spike protein of the SARS-CoV-2 gene. and it's actually working very well, and it's a very popular service. But of course, it's limited if you want to do DNA storage. I mean, because first, it's tedious to handle plasmid. You have to use bacteria. You have to do many manipulation and so on. And you just basically, you are limited to just cutting and pasting. You cannot really write new sequences and so on. So it's not really adapted to DNA data storage. And actually, it's not really adapted for many things, because in many things in DNA, in biology, you actually want to try new sequences, you want to modify stuff, you want to try things, and you are a bit limited with these kind of things. And so one of the big, I would say, breakthrough in molecular biology was the report of this so-called phosphoroamidite chemistry. So it was developed by Bocage and Carruthers in the 90s. And they came up with a way of how can we chemically synthesize DNA. So instead of just cutting and pasting DNA from nature, they found a way to actually build from scratch DNA. So I'm not going to go into the detail of the chemistry. I will just explain you the process at a high level after. But what you have to know is just basically, first, it's funny because it's completely a natural way of synthesizing DNA. The way DNA is synthesized in cell or with enzyme is from the 5' to the 3'. And all the cells and all the enzyme working on DNA, they work in this direction. But funnily, with this process, it goes in the other direction from 3' to 5'. So, I mean, it doesn't have much implication. It's just a notice. And the other thing, it's actually something that's done in organic solvent. So you don't have much water involved in this process. It's completely organic. It's based on like a classical organic chemistry. And there's not much water in this process. But actually, it's a basis of all the DNA synthesis industry now. And for example, like all the main manufacturers of DNA, like IDT, Twist, Rofin, they are all using this process. And it's very hard to overstate the importance of this process. It's just driving like much biology now, like all the PCR reaction we've heard of for the SARS-CoV-2, they're all based on using DNA that's made using this phosphoramidite chemistry. So now I will show you an overview of the process. So I found this a scheme from Twist Bioscience, which is actually very good for understanding the process without getting it into the chemistry. So this is how you make DNA when you do chemical synthesis. So you start from a blank state. So basically you attach your DNA to a solid support. And then you start by adding bases. So the first step is coupling. So let's say you want to add an A. So you put your block A that will click on the block below, and then you have your A. So then something can happen. I will go into the detail for the capping. So you click your block. Then you do a step of oxidation because usually the species you have is not very stable. So you need to stabilize, so you do some oxidation. And then if you want to click another block, you need to deblock this stuff like deprotect, which is something very classical in organic chemistry. And you remove the protection so that you can bring another block. And then you can add, for example, after the A, you can add a G and another G and so on. And in this way, you can make just any sequence provided you have enough material, enough time. So there's another step which is called capping, because actually, because it's a chemical reaction, typically the yield are not 100%, and sometimes the coupling will not work. So it means you want to put a, but the A will not click, so you will have a deletion. And sometimes you can choose to continue with this kind of sequence and say, I can tolerate deletion, and that's fine for me. But sometimes it's not tolerable. So what you do is just cap, and you say, if I fail to add this A, then that CN for this chain is not allowed to grow anymore. And you do a cap. And you see from the shape of this cap that basically you cannot put anything on top of that. So typically, this is usually it's done when you do, for example, when you order DNA on the internet, you have the capping. And so it means it's important from a point of view of coding or DNA data storage, because it tells you now the kind of errors you can have. So typically, the only errors you can have is a truncation or deletion. So if you don't do the capping, you can have deletion. So for example, just imagine you had the A, but for example, this G was not clipped well, so you missed this guy. So it's a deletion. And if you use the capping, then basically, if you failed to couple, you just stop going. So it's very important because it means if only the full size DNA will have the correct sequence. So it means just by looking at the size of the DNA, you can know if they have a good sequence or not, because only the DNA that have full size will have the correct sequence. It means you don't have, for example, insertion, you don't have mutation, which are the kind of things you can find, for example, in cells because you have mutation, for example, due to UV or chemicals. But typically, in a chemical DNA synthesis, you don't have this kind of errors. The only way you have is just to truncate your sequence. So usually, what you do, you make your sequence, and then you just purify, so you keep only the longest strand, and so you are kind of sure you have the good sequence. And it's very important because you have to remember that people in biology, they really want to have the exact sequence down to the right base pair, and that's why they need to have a process where they can be sure, pretty much sure of what they have in the step. The other thing you have to have in mind, it typically takes about 200 seconds per step. So it might not look like a lot, and you can make, for example, like 20 to 30 bases or 100 bases in one day. But actually, if you want to do DNA data storage and so on, it's actually very long because it means it takes about 200 seconds to add one nucleotide, which is about 2 bits. And it's actually not a very high throughput. So, and now this process, it's been optimized for now 40 years. It's really, really mature. And typically, you have, for example, like this is a video from IDT, like the main manufacturer of DNA. And they literally have farms of DNA synthesizer, like they have a warehouse filled with a DNA synthesizer that are just producing DNA all day and all night. The process is highly automatized. The purification, the analysis, and everything is automatized. So it's really powerful. And for example, typically, if I order DNA on Monday, I can have it on a Tuesday or Wednesday if it's going well. And in addition to the synthesis, they also will characterize the DNA to see if it's pure. And they can also do other processing. So it's really, it's really efficient. Most people now, almost nobody is making its own DNA. Everybody is buying it on the internet because it's just much simpler, because they have this scale, this advantage of scale. Like when you have this kind of a scale, you can make DNA very cheap. And very interestingly, if you look at this machine, you can see like each machine has 4 bottles, because each bottle will add one precise nucleotide. And basically at each step, you will draw a reagent from one of the bottles. And it's very, as I say, it's very hard to overstate the implication of this, because it's actually driving most of the modern molecular biology. So if you just want a few examples of the application, of course, we have the molecular diagnostic. So for example, when SARS-CoV-2 emerged, we needed some primers. So primers are DNA strands that are used to detect the RNA from the virus. And you can just make them in a few days. So when the virus emerged, in a few days, people had designed the primers, and they had synthesized them to detect them. So it's, and all the PCR or everything is based on the fact that you order some primers from a DNA company, and then you take your primers, you mix with enzyme, you put the sample from the patient, and then you will look if you get a positive or negative result. The other thing that is going also is a bioproduction. So the idea is, as I said before, you can actually insert a gene in a bacteria, and the bacteria will express this protein. And because we are made of protein, you can imagine that it's quite important to be able to make proteins for many reasons. For example, if we eat proteins, like if we eat meat, it's protein. So it means if you are able to actually control very finely the DNA, the sequence, the way it's expressed, and so on, you can actually make anything. So that's why you have, for example, a dinosaur. And for example, Ginkgo is a company that aims to just grow things based on, you have a software that is DNA, you put this in cell, and the cell will produce what you want. And it's really recently been publicly introduced on the New York Stock Exchange. And it's actually, it's a company that's a startup from MIT that started with an undergrad and graduate student working in synthetic biology. And they just use DNA all the time, and it's a basis. And it's also the basis of, for example, much of the biotech industry. As I say, like some drugs are based on for example, expressing some proteins in a bacteria and so on. And one of the last that is maybe not so famous, but that is close to my heart, is DNA nanotechnology. And actually, it's a field that emerged 30 years ago, and people realized that basically you can use the self-assembly of DNA to actually build, shape, and program things at the nanoscale. And maybe one of the most famous example is the DNA Origami that was reported by Paul Wasman 15 years ago now, just when I started my PhD. And he showed that basically, provided you can synthesize the DNA strand you want, for example, just with 200 strands, you can fold DNA to have any shape you want. So for example, he folded this DNA origami that is about 100 nanometer in length. That is very small, using only 200 strands. That he basically he said to his computer, I want to make a smiley. Then the smiley computed the DNA strand that he needed. He ordered them on the internet, and he got them. He put some salt, some water. He just heat it up and then cool it down and look under the microscope, and it got these beautiful faces. And again, this is entirely based on the fact that you can order DNA on the internet with just any sequence you want. And people in this field, they just order DNA all the time. Like it's like writing software. They order DNA on Monday, they get it on Tuesday, they do the experiment on Wednesday, and if it goes well, they can. If it goes bad, they can order again on Thursday and do another experiment on Friday. So it goes very quickly. You can have software that help you to design DNA and so on. So it's very mature. So now there's still a difference between what we want to do for molecular biology and DNA data storage. If you remember, when you order DNA from the company, you don't have one strand, you have a mix of strands. Usually it's not pure. But if you're doing biology, you really want, what you need is just one strand with a perfect sequence. So usually people, they go to a great length to actually find that strand. The way they will do that, they will actually inject the DNA in bacteria, and they will pick up the bacteria that will behave as they expect. Because if the bacteria doesn't have the full length DNA, usually it will not do what you want. And that's how they pick up the good DNA. So it's called like colony picking and so on. And so imagine if you have a mix of DNA, like maybe one will have the good and perfect sequence, and you will have lots of truncated product. And usually if you're a biologist, you don't care about those guys. You actually don't like them. You want to remove them. And what you just need is one strand but perfect sequence. Now, DNA data storage is actually very different because you don't really care about having a perfect strand, and you know better than me. Like if you have like noisy signal, but if you have enough redundancy, you can actually average things. You can get information, and you can learn a lot. So I'm sure if I give you a lot of truncated strand and so on, you can actually recompose and the original sequence by doing lots of advanced mathematics that I'm not going to teach you. But the idea is that actually you have lots of information, because when you synthesize DNA, you have thousands, millions, billions of strands. They are not all perfect, but they all carry some information about the sequence. And I think that's one of the new, I would say, thing that is emerging with DNA data storage is actually we don't have the same constraint that we used to have on DNA synthesis. Like basically DNA synthesis, it was done to actually do perfect sequences, but we don't need that for DNA data storage. And so if you look at the issues with chemical DNA synthesis, typically deletion and truncation, they're not a big deal because you can correct them with redundancy and decoding, but we have other problems. So one problem is usually the length is limited to typically 100, 200 nucleotides. It's slow. It takes a lot of time to add one nucleotide. It's quite expensive. Like if you want to code 1 megabyte of data, it's quite expensive. And you also generate a lot of waste, like a chemical waste. And typically if you ask, but then why can't I make a very long DNA? Because I just need to keep adding, because actually the yield is not perfect. And the coupling step, it's good, but it's not perfect. So typically it's something like 98, 95%, 99%, sorry. And so it means it just goes down exponentially. So every time you add something, you have 99% chance of going to the right length. But if you keep doing that for hundreds of steps, then at some point you will just have nothing left in the solution. So for example, I took this graph from IDT, and they compare the number of full-length DNA in your solution against the length. What you have is an exponential decay, but it will depend highly on the yield. So if you have 99.6, then you go down a bit more slowly. If you have 98.5, then it's quicker. And then usually you have a cutoff, and usually in the industry, the cutoff is 30%. So you say I must have at least 30% of DNA in my solution that is full length. And so once you have this cutoff, it gives you the maximum length. So for example, if you have this kind of yield, typically you will have even less than 100 nucleotides. If you have a better yield, like you can have like 300 nucleotides. But it's very hard to go much higher than that. Like this process has been optimized for decades and decades, and you cannot really shave off much more than that. So now, typically, you will not get much more longer just by working on chemistry. And usually, it's kind of the limitation now. You cannot really do longer than that unless you use different chemistry. And I'm going to talk about that. Because actually, if you remember, I mean, if you biologically see it, the genome, the DNA in our body, in virus and so on, they are very long. They are not 100 or 200 nucleotides. You just don't do anything with 200 nucleotides. And if you just take a virus like SARS-CoV-2, it already have 30,000 nucleotides. If you take a bacteria, it's more like millions of nucleotides. And if you take the human genome, it's like 3 billion of nucleotides. So it's quite a very big. And then you can ask, but how come the living organism, they can do very long DNA and we can't do it with just chemical synthesis? And the magic answer is because they use enzyme. Like enzyme is a protein, special protein that will catalyze reaction that's been like evolved over billions of years. And that is much, much better than what we can do chemically. Like enzymes, they have like amazing yield. They can do amazing stuff that we can just dream of in terms of chemistry. And that's why people now are thinking about using enzymatic DNA synthesis. So instead of using a chemical synthesis with just organic chemistry, they say maybe we can use actually an enzyme to grow our DNA. And this is, for example, one of the examples. So you have your chain of DNA that you want to grow. And let's say you want to add a new nucleotide. So you have this enzyme that is a polymerase and that carries the letter you want to add. Then this enzyme will recognize the end of this DNA. It will just like click to it, and it will add the DNA with almost perfect yield, and then it will be liberated. So you still have protection and deprotection because people don't control still very well this system, and that's a problem. But in principle, the yield are much better. So in principle, you can make much longer sequences. It's almost like 100% in principle. It's also much faster because the addition of the nucleotide can be much faster. And you can work with water solvent, aqueous solvent. So that's also a big advantage for the environment. But now you have to see like this was reported like maybe 5-10 years ago, this kind of synthesis. So it's still not that mature compared to chemical synthesis. And in practice, it's still not beating the chemical synthesis, but there's a lot of margin for progression. And probably we will hear a lot about this in the future. And for example, like in the project we have with Mark, we plan to use enzymatic DNA synthesis to do a better DNA data storage. And actually, that's my expertise using DNA enzyme. I'm more an enzyme guy than a chemical guy, because actually enzymes are just amazing at working with DNA. And for example, like there's a French startup, a French company, DNA Script, that is leading the market now. And their plan is actually to make the equivalent of the personal computer, but for molecular biologists. So the idea is like you would buy this machine, it would sit in your lab. And when you need to make some DNA code, you just take your computer, you write your sequence, and this machine will make it overnight using enzymatic DNA synthesis. Because actually you don't have all these nasty and toxic chemicals. It's actually much simpler to operate, and you can have it in your lab. It's more complicated to have a chemical synthesizer in your lab. You can have it, but because you have to handle all the waste and so on, usually people don't like it. And that's why actually they prefer to order it on the internet, because they don't have to handle all the toxic waste. So it was just released in June, so it's still a new product, but they hope that people will just buy this kind of personal computer for biologists, and you can just print the DNA you need and get it overnight. So it might be actually a new way of working with DNA synthesis. But actually, the throughput, as I say, is not very good. So it means if you want to store information, it's not that much because the speed is not high. So if you look at the throughput, it's a factor of time and speed. So the speed is how many nucleotides you can add per second. So how long it takes to add one nucleotide. And here it's based on the chemistry, and you cannot really get much more. So typically it will take 1 minute, 2 minutes to add one nucleotide. And it's been optimized for now 40 years in the case of chemical synthesis. And it's really hard to gain many orders of magnitude. But there's another parameter, which is space, because you can do all this reaction in parallel. So if you find a way of making many sequences in parallel, then you can gain a lot of throughput. And actually, it's just based on the idea that electronic was based on the idea that if you parallelize stuff and you have many, many transistors in parallel, you can have huge gain in terms of computing powers. It's the same thing like CPU. You can either increase the frequency of the CPU, but it's really hard, it gets hot and so on, or you can just have many, many more transistors. And people are moving in this direction for DNA synthesis. It's hard to increase the frequency of the synthesis, but you can do many, many more reactions if you can miniaturize things. And just as a reminder, like the Moore law, it's the idea that you can double the number of transistors that you put on a chip every 2 years. And it has been almost going like this for 50 years, 60 years. And you can now put like something like 50 billions of transistors on a chip. And the idea is like, if you can use this miniaturization, this kind of process of miniaturizing things, and if you can actually miniaturize the synthesis using electronics, then you can gain the same order of magnitude. And for example, in this plot, it shows the cost of DNA synthesis here in red. So you have two kinds of synthesis. So the DNA synthesis, a gene synthesis is when you have a full gene, and oligosynthesis is when you just order a small strand. So usually when you make a gene, you order the small strand, and then you have to stitch them up together to make a full gene, because you cannot make a long DNA, as I said. And typically you see the cost, it's going down, but not that much. But now if you look at sequencing, so sequencing is, I give you a sequence and you have to read it. And there's been a huge drop in the cost of sequencing. So you see it was going down. And then something happened around 2005, when actually they used, they miniaturized sequencing using optics, microfluidic, and so on, and using new chemistry as well. And they get a huge drop in cost. And now it's been a huge revolution, because for example, for the human genome, it cost before like maybe $3 billion to get it. Now you can do it maybe for $100, $1,000. And this is all based on the fact that you just miniaturize reaction. And so actually you can do the same thing for synthesis. So there are several methods. The idea is you do the synthesis that I showed you before, but on the surface. And one idea is, for example, you can do inkjet printing, like the same that you use to print stuff. Instead of printing ink, you will print A, C, GT, and so on. So here you will be limited by the resolution of your ink of your head, and so on. And actually, the problem is hard to manipulate. When it gets very small, then you have to manipulate really many liquids, and it's complicated. The other method is just electrochemical synthesis. So imagine you have some transistor and some electric field. And actually, it's very easy to control electric field with electronics. And you will, by using electric field, you can control the pH. And if you control the pH, you can control the synthesis somehow. So it's also a method that is now kind of mature. And the last method is just a light-directed synthesis. So you will send an image, like you send an image on your surface, and where you have light, but it will deblock and unprotect some reaction, and you can, for example, add a A. And when there is no light, you can do nothing. And this, for example, this is an example of an electrochemical chip that has almost 100,000 spots. And in parallel, you can make about 100,000 oligos. And just to give you an idea, like people now, they are thinking about massively miniaturizing things to bring down the cost of DNA synthesis. So now the cost of DNA synthesis, typically, if you parallelize things, it's about $1 per kilobyte. And they hope that in 4 or 5 years, by doing miniaturization, we'll reach $1 per gigabyte. But because the Moore law has been so strong for so long, people expect that by the end of the decade, we'll be able to miniaturize so much, like put more than like 1 trillion spot on a surface, that it will cost only $1 per terabyte. And when you reach this kind of cost, then basically we can change many things because it's even cheaper than some hard drive now. Okay, so that's the end of my presentation. So thank you for your attention. If you have any question, I take them. Thank you.
Thank you, Anthony, for this very nice presentation. So very clear. So it's, I think people here appreciate. So if you have questions.
Yes, I wanted to ask a question actually.
Yeah.
Because I was not aware about all the chemical waste that you have when you are synthesizing DNA. And actually one of the claims for DNA data storage is to avoid all the pollution produced by data centers. So I wanted to ask if there is some study or estimation about the impact of DNA synthesis.
Yeah, it's a good point. So actually, yeah, so I think actually there's a lot of waste because they use something that is called acetonitrile. And because the way when you do synthesis, actually you put one nucleotide and you wash everything. So you lose a lot by washing. And actually some company, DNA synthesis companies, actually they recycle the waste because it's so precious, and they distill it harvest and start again. But the thing is, because actually, if you want to scale for DNA data storage, it would not be environmentally friendly. Like now, the company, they're not doing much data. In terms of data, it's not so much. Like if you want to do even 1 megabyte of data, it will be huge in terms of impact. For biology, it's okay because you don't need lots of sequence and so on. But if you scale it, for example, to 1 gigabyte, it just would be not possible. It would be huge in terms of impact and so on. And that's why one way to do that is actually to miniaturize. So if you miniaturize reaction, then you need less reagent. So it's one way of reducing waste. And the other way is actually to use enzyme and to work with water. And that's why there's a lot of interest in that. But the thing is, the thing I showed you before, like the DNA farm with synthesizers, it would not just be possible. That's why you need to miniaturize, because it would not be environmentally friendly. But typically, people don't talk so much about that because it's not. And even me, it took me some time to realize that it was done like this. But yeah, it's kind of actually kind of nasty chemicals if you are doing the chemical synthesis. Yeah.
And then in that case, Anthony, synthesizing DNA will be the pollution of doing this will be higher than the pollution of data center.
Yeah, to be honest, I didn't compare. It's really hard to compare because it depends on how you do it. I guess if you can miniaturize things, it can be very small, because if you can miniaturize on the surface,
if not,
if you don't, I mean, it's just not possible, because then you need so many synthesizers and so on that it would be huge. It would be huge, and I think people are not even trying. So this kind of bulk synthesis, I think nobody thinks about it for DNA data storage.
Okay, so things that Twist are doing are not
exactly. So Twist are not doing this bulk stuff. They are doing miniaturization, and they are the pioneer in this kind of stuff, because they miniaturize the reaction, and actually they are pioneer in this kind of stuff. So they are very advanced. Are using kind of advanced inkjet printing and so on, and are using microfluidic and so on, because it would just not be doable, I guess. That's why actually you cannot do things with Twist that you cannot do with other companies, because they have this expertise.
Okay, so thank you. Touraj, you have a question?
Yes. First of all, thank you, Anthony. Very nice and informative presentation.
Thank you.
I have a question. I hope I didn't miss it, but if I understand, the way things are progressing, the issue of cost is going to be resolved in terms of storage of large amount of data.
Yeah.
What is the prognostic for the future in terms of throughput? So I understand that there are ways to parallelize, etc. But let's say the day 1 terabyte will be $1, let's say 5 years, 10 years from now, how fast this can be done? Like how long does it take to have 1 terabyte?
That's a very good question. I think it could have happened a long time before. It's just because there was no market and no user. And because like all these technologies, the Moore law scaling and so on, they have been available. It just, I think that there was a need to show there are users and people interested in DNA data storage. And now like you have people like coalescing around this goal, and more and more people are interested. And now you see investment from government, either for example in US or France or other countries. And what people say is like you need maybe to put $50 million, $100 million of investment in it to make it happen. But usually people that are kind of, I would say, kind of very optimistic. And if you look at the curve I showed, people say that in 5 years you can do it. It depends on the chemistry of what you're using. But for example, Twist or other stuff, they say we use the same chemistry, just miniaturize a lot. And you cannot deny that the electronic industry has been very good at miniaturizing things. So if they get on it and if they get the investment, because typically you will need to work, with a foundry and so on, because it's very heavy in terms of electronic. But people think that in, for example, 5 years, you could cut down the cost by 1,000. That's realistic. So I think that's a thing. And then once you cut the cost, maybe you will have like some early adopters, some early users that are really interested. Maybe for example, government, archive, and so on. And once they are in, then the idea is it will bring more customers. And so it will help to have more investment, and so it will bring down the cost. It should be like a virtuous cycle. That would be the idea. But the thing you need at some point now, it's just too expensive, and we need to overcome the initial cost. And once it's cheap enough, people will come. That's the idea now, yeah, I guess. So in terms of time scale, having people are, for example, like the MIST project that was started by the YAPA, I think last year, and they aim to bring a reagent cost of about $1,000 for 1 terabyte before 2025, to give you an order of magnitude. But that doesn't mean you will have to pay this cost, because then it's just the early development. But you can imagine that a few years down the line, it will be available. So, I mean, the time scale will be typically 5-10 years to have something that's realistic and usable for users, I would say. That's the time scale, yeah, I guess.
Yeah, but I was actually talking about the other time. So I understand between 5 to 10 years, the cost will be low enough so that it becomes competitive with other alternatives like tapes.
Yeah.
But then how fast, like how much time does it take?
Okay. Sorry, so I didn't understand the question. Okay, so yeah, if you look at the YAPA, they aim for 24 hours. So they have a metric, which is to say you have to encode something like 1 terabyte in 24 hours using like something 1 square meter. So, and typically, so they're aiming for hours or days. That would be the idea. And typically you cannot do minutes or seconds because you have mechanical stuff and so on. If it's weeks, it's just too long. What they're aiming for is typically you can encode DNA in 24 hours and you can read it in 24 hours. And if you are doing like a cold data archiving and so on, this is not really inhibitor. I mean, this is something you can tolerate, I guess. Hours or days, that will be the time scale. So of course it won't compete with SSD or very fast access, but for cold data archiving, like archive, days or hours, it's something you can tolerate, this kind of stuff.
Thank you.
Thank you for the question.
Sorry. Okay, thank you. Other questions? No. I have a question, Anthony, about the length of the oligo that can be generated. It seems impossible today to synthesize a long oligo more than, let's say, 300 nucleotides without errors. So why really it's not possible? Because
actually, even without error, the thing is you have exponential decay. So each time you want to add a new base, you do it with a success rate that is not 100%. And so if you have 99% chance of going the chain, then basically you can imagine after 100 rounds, then basically you will not have many people that are gone. So the thing is just imagine you are flipping a coin. 99% of the time it's right, but if you get it wrong once, then it's finished. This chain is not going. So that's why you are limiting. So if you are starting from big pool, then basically you can do 200, 300, and then you can purify. But typically for what people want to do, they want actually, like typically 30% of a full length. So that's why it sets the stuff. So you have exponential decay, you have your yield, and then you just take the logarithm to know the maximum size. And if you want to, for example, to get 10 times longer, you need to have 99.9%, which is very hard to get. And if you want to get even 10 times longer, you get to 99.999. So it's typically because the yield, 99%, it could look like it's big, but actually it's not that big if you are doing very, very long stuff. And for example, if you look at a polymerase, they are much better. Like they can add maybe 10,000 nucleotides without errors in one go, which is much longer than what you can do. And then you have many processes actually to replicate DNA in cell without problems. Like in your cell, you have 3 billion of nucleotides that are replicated without any problems. It's just because the way we do it is very crude with chemical synthesis, and if you fail to couple, it's finished. So
okay, thanks.
Sorry, but then with enzymatic synthesis, the length wouldn't be restricted anymore to a couple of hundreds.
Yeah, so that's the idea. So people, so ideally now we should be to do much better. So now it's still starting, and if you look at the length of the best DNA enzymatic synthesis, it's about the same than chemistry. But it's because the field is very new and people are expecting they will be able soon to do very long. So typically they're aiming for 1 kilobyte of, 1 kilobase, because it's typically the size of the gene. But with enzymatic synthesis, in principle, you could do much better. It's just now it's very young, so they're not that long, but they're going very fast. For example, DNA script has been breaking records and records in terms of length. And I guess maybe the best they've done now so far is 200 nucleotides, but maybe 5 years ago it was maybe 20 nucleotides. So it's going very quickly.
Yeah. And is there theoretical limits to that?
No, I mean, in principle, there's no theoretical limits. I would say typically would be, you cannot really, I would say 10,000, because for example, like some polymerases that are very possessive, they can do 10,000, 20,000. So then you have problem. But I think if you can do it, still 100 times better than the chemical synthesis. So but I don't think you will make a genome in one go. A genome is like 3 billion base pairs. But if you can do 10 times or 100 times better, it's still much better than what you can do now. Yeah.
Okay.
But then the constraint will come by the manipulation of the strand, because the longer the strand, the easy is to break it, right? When you are
so the thing is, when you get longer, you have many other problems that are coming in and so on, and then you have many bottlenecks. So that's why you will be limited by other things. But that's the point. For example, if you want to make a gene, typically they have these pieces of 100 nucleotides and they stitch them together with enzyme. It's very tedious. So if you can get 1,000 nucleotides in one go, you have 10 times less steps and so on. So that's why people are expecting it will be more efficient.
Okay.
Thank you.
Last question, maybe to Raj.
Yes, it's a question that comes, Anthony, many times in the past in the workshops and discussions we had in JPEG DNA Atto group. So if you can make longer genomes, much more than the short ones today, do you think there is a danger of synthesizing viruses or create a biohazard, either knowingly or unknowingly, especially for maybe not storage necessarily, but for markers? You know, people use also DNA for markers and tracing and things like that.
That's a very good question. So we have to think about that. Some people think that you could, you could go to the scene of a crime, you could synthesize the DNA of somebody and put it on the scene of the crime and show its him and so on. So I think if you can synthesize very long DNA without errors, then probably we have to think about many new things. But typically, it's already a problem. For example, if you order some DNA from IDT, they ask you, does it come from a virus? Is it dangerous or so on? Because you can ask IDT to stitch this DNA together and to have a long DNA. They can make a gene for you. And when you order on the internet, you have to certify you are not making a virus or so on. But the thing, as I said, if you really want to make a virus, you can actually take one from nature and modify it. Like it's a cut and paste and cloning. And it's simpler than making it from scratch. But of course, it's a big problem. So the thing is, I think with DNA data storage, because there will be many errors. We're not trying actually to be genetically correct in terms of code. So I don't think the DNA that will come out from the DNA synthesizer for DNA data storage will be dangerous, because the way it's done, you see, we can tolerate many errors. So maybe there's a lot of truncation, deletion, so it just will be useless. But who knows, like if we get really good technology and you can get very long DNA with a very long in and so on, then that would be a question. But people have been, they've been asking this question for now 50 years, because when people started to be able to manipulate DNA, then basically they say, oh, maybe we should have some moratorium and think more about this, and they put some, I would say, guidelines in place, because they've been worried about this question for 50 years or 60 years now, and now it's becoming more and more real. And with CRISPR-Cas9, you can even edit the DNA, and you can edit the DNA of human and so on. So I mean, it's posing a lot of ethical questions, and I think it's important to not ignore them and to think about it in advance, if possible, not after the technology has come, because the technology can arrive very quickly. So if you have not thought about that before, then you are kind of in trouble. Yeah.
Okay, so thank you, Anthony. Thank you for this nice presentation. So now the next talk is efficient synthesis of DNA via cost-constrained systems. So the talk is given by Andrea Lenz. So Andrea receives the Bachelor of Science and the Master of Science degrees both from high distinction in electrical engineering and information technology from the University of Technology in Munich, Germany in 2013 and 2016, respectively. In 2014, he received the Leo Brandt Award Master of Navigation from the German Society of Positioning and Navigation. And since 2016, he is working as a doctoral candidate at the Coding for Communication and Data Storage Group at the University of Technology in Munich, where he's involved in research about coding theory and insertion and deletion errors and modern data storage system. So, Andreas, so I give you the floor for your presentation, so 30 minutes max. Do you have some problem, Andreas?
He's muted.
Yeah, you are muted, so maybe if you can.
Yeah, sorry, my Zoom
you have...
disappeared. I'm very sorry about that. Give me 1 second.
Yeah.
Okay, can you see my screen now?
Yeah, that's fine.
Okay, I'm very sorry about that. Okay, so thanks a lot for your presentation. And also thank you very much for the invitation to this nice workshop. I'm very glad that I can present. And also thank you very much to my previous speaker, who actually introduced some of the topics very nicely, which I'm also going to talk about. So first of all, we are a group of coding theorists and information theorists from Technical University mainly, but also from University of California, San Diego, and from Technion in Haifa. And we are basically working on on problems that are motivated by DNA-based data storage. And we're trying to abstract and optimize them using coding theory and information theory. And more recently, we were also joined, or we got support from an expert in combinatorics, Steve Maxwell, who's helping us with our work. All right, so let me shortly give you an outline of what I'm going to talk about. I want to talk about the system model that we're going to look at, the problem formulation, and then cost-efficient DNA synthesis. And we will show that how they relate to subsequence counting problems and also cost and graphs. And finally, we will relate them to cost constraint system, which will help us to actually come up with solutions to the problem. And finally, we're going also to look at the case, what we call constraint synthesis, and I'm going to tell you later what it is. Okay, so from an abstract point of view, what is data and storage in DNA? So usually, we have some binary data that we want to store in some archive. And in DNA-based data storage, we use some appropriate encoding mechanism such that we get a bunch of DNA strands. Then we send them to a synthesis company like Twist, as we heard before, or maybe even we can do it ourselves in our lab to create these DNA strands by ourselves, which means to synthesize them and then put them in an appropriate storage container. And finally, if we want to access the data, we're going to sequence the DNA, which gives us some possibly erroneous version, as we heard before, of the original strand if something goes wrong. And then we may want to decode the data such that we get back our original user data. So what we're going to focus on this talk is to optimize the DNA synthesizer. As we heard before, synthesis can be a very costly step in terms of both actual costs and also in terms of time. So what we are going to focus on is something that we call array-based synthesis, which means that the DNA strands are synthesized by a nucleotide fashion. And for each nucleotide, we're going to flush the nucleotides and select a bunch of strands for which we are going to attach the nucleotides to a specific selection of strands. So maybe let's look at the following example. So assume we want to synthesize 3 strands, CT-ACG, AGTA, and CTT, which we got by our encoding mechanism. And now we take these strands, send them to Twist, or synthesize them by ourselves as follows. So first, we choose a synthesis sequence, which in this case is ACGT, ACGT. And we're going to cycle through the sequence and build our 3 strands based on this sequence, which means we start with the letter A, and we can attach it to our second sequence because our second sequence starts with an A. So we attach A to our second sequence. Next, we attach C, which we can attach to our first and third sequence because they both start with C. And then we cycle through our synthesis sequence, and in each step, we attach the nucleotide whenever we want to attach it, such that we build our correct strand. So here we can attach G. Now we attach T and for all of the strands. Next, we attach A for the first 2 strands. Then we attach C for the first, G for the first, and T for the third. And as you see, we have built the 3 strands correctly by cycling through this synthesis sequence over here. Now, I hope that was illustrative, and I want to go a little bit more mathematical description of the problem so that we can actually formulate an optimization problem that we can solve. So assume we have a bunch of DNA strands, X1 to XK, that we want to synthesize. And as we heard before, this can be many in parallel. So maybe 10,000, 100,000, such that the overall speed is actually fast, because we also heard that one step in such a cycle can take up to 1 or 2 minutes. So we want to do many of them in parallel. And of course, so the alphabet that these sequences are here is Q equals 4, because we usually have 4 different bases for DNA. Now, we choose on a common synthesis sequence S, which we saw before was the ACGT, ACGT. And we can define something that we call synthesis time. And this synthesis time is a function of the synthesis sequence S and the strands that I want to synthesize. And it is defined as the minimum T, so the minimum time, such that X, so all the sequences Xi are subsequence of my supersequence S1 to T. So in this case, I can synthesize all of my strands if this is given. If I choose as many synthesis cycles as the synthesis time tells me here. So for example, as we have seen in our previous example, that if we choose the synthesis sequence ACGT, ACGT, and we have those 3 strands, then we end up with a synthesis time of 8, because we could synthesize them all after 8 cycles through the synthesis sequence. Now let's go to the mathematical description of the problem. And now, as you might notice, there are some strands which take longer to synthesize and some strands which take shorter. So for example, here CTT takes very long, although it's short, but CT-ACG takes short, even though it's longer. And therefore, the idea is to enforce an encoding such that we do not allow all possible DNA strands to be synthesized. And this is what we call a synthesis code. And mathematically, this is just a subset of sequences over the nucleotides ACGT. Now, more mathematically, what we are actually trying to focus on in this work is we want to maximize the information that we can synthesize in time t, which means basically that if we look at the largest code such that the synthesis time of this code is at most t, this is what we call NST. Okay, and I want to go through a short example that illustrates this here. So think of it as follows. So we have a synthesis sequence S as ACGT, and we have 4 cycles. So we will allow 4 attachments of nucleotide. So what is the maximum number of strands that I can synthesize in 4 cycles? So first of all, I can synthesize A, C, G, T alone. Then I can also synthesize AC, AG, AT, CG, CT, or GT, right? Also, I can synthesize length 3 sequences, ACG, ACT, AGT, and CGD.
and finally ACGT. I can synthesize all of them, which means that I have 15 possible different DNA sequences that I can synthesize, and which gives us, if we take the logarithm of this number and divide by 4 cycles, which tells us that we get roughly 0.98 bits per cycle. And notice that, for example, we cannot synthesize something like AA or TT because we are restricted to have a subsequence of ACGT that we can synthesize. Otherwise, it would take more cycles, and that's what we want to avoid, right? So another example is, I mean, if we choose a different synthesis sequence, which is obviously not as good as the first one, the best that we could do in this case is to choose the codebook. So either we synthesize AA, AAA, or AAA. And I know this is not very practical, but I just want to highlight that actually this results in a smaller synthesis rate, because in this case, we can only synthesize 0.5 bit per cycle because we have 4 code words. We take the logarithm and divide by 4, which means we can synthesize half a bit per cycle, which is of course worse than in the first case. We want to have like synthesize many bits per cycle. Okay, now with that, we arrive at the central definition of this talk, which is the synthesis capacity. And it is simply taking log the number of sequences that I can synthesize, divide by t, and let t go to infinity. And this tells me basically, sorry, I can also define this for fixed length sequences, because in many cases, we don't want to have different length sequences, as we had heard before also. So here, small n is a parameter which controls the length of the DNA strand. So maybe this number now tells me if I have T 200 and 100, how many sequences can I synthesize which have length 100 and take 200 cycles of DNA synthesis. And we will define the fixed length capacity as follows, that we control the length of the sequences by this parameter alpha, which is just a value between 0 and 1. And we also take log and let t go to infinity. So intuitively speaking, what this capacity tells us, it tells us how many bits can we how many bits can we synthesize per cycle. And this is what we want to maximize. Now, what we did in our work was to characterize this capacity for both the variable length case, which was the first one, and also where we want to have fixed length sequences. For periodic sequences, so for arbitrary periodic sequences, in fact, we show, as I mentioned before, how they relate to cost constraint systems, which also allows us to actually build practical codes in the end. And finally, we shows a proof of how to compute the capacity for arbitrary constraint systems, which then gives us the capacity of the synthesis process. All right, so let's shortly review a bit of the related work. So there's definitely more, but I'm only going to highlight that this is most related to ours. So Jane et al. Showed last year, they discussed a little bit different type of synthesis process. And this is terminator-free template independent synthesis. And this is a process where actually the run length of homopolymers that you add in every step is not always 1, but it's a random process that you can control. So it's a little bit more erroneous process, but it is a little bit cheaper and therefore allows for possibly allows for like higher cost efficiency. But it's a different process than what we are discussing, and also what was discussed in Makarichev 2021. They discussed batch optimization, which means that if you have many DNA strands, so say you want to synthesize 1 million DNA strands differently, this is they discussed on how to distribute them into different groups such that you can perform an efficient synthesis on each of these groups. Now, from a technical perspective, like one of the most relevant papers to ours is that famous paper by Shannon in 1948, which introduced cost constraint systems and also showed connections to Markov chains. And finally, more recently by Zoriaga et al., it was shown how the capacity of fixed length cost constraint system
looks like and how it relates to probabilistic Markov chains. And finally, also from a technical point of view, which we use for our results, is the results on analytic combinatorics and several variables, which have also found recent interest by experts in combinatorics, for example, by Pimentel and Malexia. Now, all right, so let's look at how actually this problem translates into a graph theoretic problem or constraint, cost constraint system. So now let's build a graph from the synthesis sequence in the following way. We take the symbols of our synthesis sequence and as vertices, and we draw an edge to each next appearance of each symbol in the sequence. This means if we go back to our example, ACGT, we start with A, we draw an edge to the next C, the next G, the next T, and the next A. So these are our edges. Now we do the same thing starting from C, we draw an edge to all the next appearance of the symbols, and we do this for all the symbols. So this is our subsequence graph. And in fact, it's actually not so hard to observe and also not much harder to prove that a sequence X can be synthesized if there is some path through this graph that actually generates my sequence, because this is naturally how the synthesis process works, right? And also from a mathematical point of view, we can show that the number of sequences that we can synthesize in time t is exactly the number of paths through this graph. So we have transformed this problem into a graph theoretic problem. And we can even simplify it a little bit further if we look at periodic sequences. So usually what is being done is that you use a periodic synthesis sequence. This means you have a sequence S, and then it consists of like a small period of length L. So for example, here we have a sequence AAC, and it has period 3 because we keep repeating AAC. And this is the subsequence graph, as we have discussed it before, of this sequence. Now, this sequence actually, this graph, sorry, it can be simplified by folding the graph into a graph with 3 vertices. And what it has the symbols of one period as the vertices, and the edges are the same as in the original graph, but we assign them with weights, and the weights are exactly the weights, the number of symbols that I'm skipping within this edge, basically. So here, from A to A, we have an an edge with vertex with cost 1, because we move on by one symbol. Then from A to C, we have an edge with cost 2 here, which has cost 2 because we moved on by 2 symbols. Or similarly, also from C to C, we have an edge with 3 symbols because with weight 3, sorry, because we had to move on by 3 symbols to go from C to C. So we have built a costly graph, a costly directed graph for a periodic synthesis sequence. And as it turns out, this graph nicely describes actually the quantity that we're interested in as a path through, as number of paths through this graph that have some weight at most t. And the edge weights here that we introduced, basically they define the costs for a specific path, and this graph here, it describes a cost constraint system, or what is called a cost constraint system. And this is simply a directed labeled graph with costly edges. Okay, so now what we have done so far was to relate our original synthesis problem to a cost constraint system. So from now on, we're going to discuss cost constraint systems. So originally they have been introduced by Shannon, and they are defined by a strongly connected directed graph that has some vertices, some edges, some directed edges, and an edge labeling, which is deterministic, which means that all the outgoing edges from one vertex have distinct labels. But and the edge costs are simply natural numbers that tell us how much does it cost to traverse a certain edge. And again, to go back to our example, this is the same graph as we have seen before. Now we have also added the labels that we traverse whenever we go over a specific edge. All right, so the most important quantity for cost-constrained systems is actually the language. And the language is just simply the words that are generated as path through this graph that have cost at most t. Okay, and similarly, we can define the language size just as the number of words that are generated by these path. And with this, we arrive at the central definition for cost-constrained system, and this is completely analogous to what we have seen before, that we define the capacity just as the log the number of paths divided by t, and then we let t go to infinity. And this is how Shannon originally defined it, and we will also use this definition. And also similarly as before, we can also restrict ourselves to fixed length path, which means we only look at path of length alpha t, and we compute this limit. All right, so again, as before, this capacity is measured in bits per unit cost. For the case of synthesis, this unit cost is just the number of cycles that we need for synthesis. And we can proceed to present the capacity results for these cost-constrained systems. So for that, we need a little bit more definition. Basically, what we need is a cost enumerator matrix, which has the same size as the graph, and it consists of x to the cost the entry ij, which means going from vertex I to vertex j, has simply the entry where we sum all the x to the costs of the edges, basically. And we can define the spectral radius of this matrix as a function of x also, because this matrix is a function of x. And with that, we arrive at the classical Shannon theorem, who has proven that x0 is if x0 is the unique positive solution to rho x equals 1, then this is the capacity. And now we have extended this result to the case of fixed length sequences, where the formula is a little bit more complicated, but it follows the same ideas. So if we denote x0, the unique positive solution to this determinal equation or critical point equation, then the capacity is given by this formula. And you don't need to remember the exact formula, but I want to discuss why we have two different parameter regimes here. And one is the one for small alpha, where the capacity is simply linear in alpha. And that is the case where the length of the sequence is so small that the costs basically don't play a role anymore. And then there is the second regime for alpha, where the length of the sequence is larger and we get a concave function in alpha. So we'll also see an example later for this formula. So note that actually this theorem or a similar result has been derived by Soriaga et al. In 2006. Here, actually, we have a little bit more general result that describes the precise asymptotics of these sequences. And also, we have proven it in a different way, which allows to use the rich literature of analytical combinatorics for these kind of problems. So, all right. So let's look at an example. So that was very technical, but let's go back to our alternating sequence, ACGT, ACGT. So if we draw the synthesis graph, it looks like this. Again, we draw an edge from one to the next symbol, and the cost just corresponds to how long I have to wait when I synthesized an A, and I want to synthesize, for example, a C afterwards. So here I have to wait 1. If I want to synthesize A and then another A, I have to wait 4 cycles to be able to synthesize another A. So these are, as a reminder, the costs of this graph. Now we can compute this cost enumerator matrix for this graph as follows, just by definition. And also the spectral radius of this graph is simply this equation here. And now it is well known actually that the alternating sequence maximizes synthesis capacity due to the fact that the alternating sequence has the maximum number of subsequence, in fact. And if we compute the capacity, we get 0.947 bits per cycle, which means that, for example, if we have 200 synthesis cycles, we can roughly synthesize like, okay, now I have to compute, it's like 195 bits or something like this in 200 cycles. If we compare this to the uncoded case, what does uncoded mean? Uncoded just means I map my information to bits, my information bits to DNA symbols by a simple mapping where, for example, 00 translates to A, 01 translates to C, 10 translates to G, and 11 to T. And if we look at this case, actually, you only get a synthesis rate of 0.5 bits per cycle. And this is because in the worst case, if you're unlucky, you might have to synthesize a sequence which is AAAA, for example, and that takes very long. Again, this is not very practical because there are reasons why one wants to avoid these sequences. But in terms of numbers, we see already that coding can increase the capacity quite a bit, or the achievable information rate. All right, now we have also generalized these results to arbitrary alphabets. And okay, so the most relevant case for DNA is 4 because we have 4 different nucleotides. But if we were to synthesize molecules with larger number of nucleotides, we could also look at maybe 5, 6, 7 different symbols. And therefore, we also show these results here. But for now, for DNA, Q equals 4 is the most relevant. All right, now if we look at the fixed length capacity, so far the sequences could have arbitrary length. Now, if we look at the case where we have fixed length, we actually, I'll skip these formulas, but let's look at only these plots down here. What happens is we have for a small alpha, so what we're plotting here is the capacity over alpha, and alpha was the parameter that controls the length of the sequences. And again, we see that for small alpha, we have this linear behavior, as I explained shortly before, which means that in this case, in this regime, we actually, we can almost synthesize all the sequences that we want because the length is so small as compared to the cost, actually. And ideally, we want to, of course, operate in a regime where we have a large synthesis capacity, which is for the case of Q equals 4. So the case of DNA alphabet is somewhere over here. And we actually see that the maximum of this curve corresponds to this 0.947, which we have seen in the previous slide. Okay, now I actually, I think I will skip the idea of the proof because I'm running a little bit short of time. But what I can mention is that we are using, for our case, we're using tools of analytic combinatorics. And whoever is interested in this topic, I can warmly recommend the book from Steve, which gives a very nice overview and introduction to the topic. All right, but I want to focus a little bit more practical topic, which is the case of constraint synthesis. So far, we have just dealt with the case. Now, what happens? So how can we compute, or how much information can we synthesize per cycle when we allow to synthesize all possible strands? But often in practice, we actually have the case that the sequences that we want to synthesize need to fulfill a constraint. And I'll talk about that shortly later. But in general, we usually can represent constraints in form of a graph. And then the allowed sequences that we can synthesize are then just defined as all sequences that are generated by path through this graph. So let's look at an example. Often we want to have no homopolymers because of reasons of stability. And probably everyone here can tell me better why homopolymers are not a good choice for DNA sequences, or why they make them these molecules unstable. But here we see the graph that allows no homopolymers, which means we cannot have the same repeated symbol nucleotide behind each other. So for example, if we synthesized an A, we are not allowed to synthesize another A. We can just synthesize a C, another G, or another T, which means that we don't have those self loops here in this graph, and which means that we cannot synthesize 2 times the same sequence, the same nucleotide consecutively. Another graph, which is maybe less practical, but or nevertheless interesting, is such a graph which imposes the constraint that we can synthesize everything, but we want to have two T's followed by each other. And what I'm presenting here, our theory holds for all constraints, which means that whenever you need, from a practical point of view, you need a specific constraint. The only thing that you need to do is design this graph and then apply the theory, basically. And okay, as an example, with the first graph, we can generate any sequence where we don't have any homopolymers. And the second graph, we can also generate everything, but if we have a T, it has to occur in pairs. Okay, all right. So what is it now that I'm talking about? I'm talking about, now I'm trying to define and find the capacity of these kind of constraint systems or constraint synthesis problem. And to do this, we need a small technical thing, which we call the labeled graph product. And so assume we know what the synthesis sequence S is, and we also know how its subsequence graph looks like, and say we have given a constraint graph. So you have a synthesis sequence, you have a constraint graph, which means you are only synthesizing a bunch of constraint sequences. And then you can define a labeled graph product, which basically, in short words, is a graph product where you take the Cartesian product of the vertices of both graphs, and then only take those edges from the graph product where both the labels are the same, actually. I'll go over it in an example. But the important thing is that we can compute the constraint synthesis capacity just as the capacity of this labeled graph product. And what is the constraint synthesis capacity? It is just the capacity as we have defined it before, but we are only allowed to synthesize sequences which fulfill a certain constraint. All right, let's go through this example. So here we have the good old alternating sequence, ACGT, as our original sequence. Then this is the constraint graph that we want to fulfill, which we had before. Now, if we take the labeled product, then if we work it out, the graph looks like this, and it might look confusing. But the important thing is we can compute the capacity of this graph with the methods described before. And if we do this, for example, for the alternating sequence, as we have done it here, then we get a capacity of 0.719 bits per second, not bits per nucleotide, sorry. And interestingly, if we choose an optimized sequence, which is only ACG, we end up with a higher synthesis rate. And in this case, it's not very surprising because here, if we synthesize 2 times T, it is very costly for the alternating sequence because it always costs us at least 4 cycles. So it's more efficient actually to just skip T in this case and not use it in the synthesis sequence at all. Okay, but more generally, this tells us actually that there are constraints for which the alternating sequence is not optimal anymore. And this is quite interesting from both a practical and theoretical perspective, because we want to have fast synthesis also when we're synthesizing constraint sequences. All right, so this brings me to the end of this talk. So we've talked about time efficient synthesis in terms of how much information can I synthesize per synthesis cycle. We have shown equivalence of optimal synthesis costs and path-through graph and subsequences. So these were the technical contributions. We have shown a solution for arbitrary periodic sequences. And also, like as a technical side note, interestingly, these results also imply result for the asymptotic growth rate for the number of subsequences of a sequence. We looked at constraint synthesis in the very end. And what might be interesting to look at in this setup is, for example, incorporate error correction, because here so far we only looked at how can we optimize the synthesis time, but usually we want to have error correction, which because there can occur errors, and in the end we need to correct them. And another idea is to look at variable synthesis sequences, which might depend on the sequences to be synthesized. as which is different from our setup because we assume that the synthesis sequence is fixed and given in the end. So thank you very much for your attention, and I hope I could present this topic to you, and I'm looking forward to questions.
Thank you, Andreas, for this nice presentation, an interesting presentation, which gives some links between what people are doing on DNA and information theory. So it's very interesting for us. So maybe we have some questions, and Liu, maybe if you want to ask a question.
Yes, thank you, Andreas, for the very nice talk. I have maybe a quick question. Could you go back to the graph of the capacities you plot?
To the capacity plot, yes.
Yes. So
that one?
Yes.
Okay.
So the difference is the field size Q, is it? And what is the Q means here?
It is the field size. So it is more a theoretical investigation. What would happen if I were only to synthesize sequences which consist of 2 nucleotides, for example, if I can only use A and C and not A, C, G, and T.
Okay.
So this is Q equals 2, Q equals 3, Q equals 4. So Q equals 4 is the standard DNA case, and yeah, Q equals 2 is a case where I'm using less nucleotides.
Mm-hmm. Okay. Yeah. Thank you.
Okay, thank you. Eva, maybe you have a question?
Hi, thank you for the talk. It was very interesting. I wanted to ask you, because when you were showing the alternating sequence, it's always A, C, T, G or something like that, just the 4 nucleotides repeated over again. But is there any way that you can modify this alternating sequence to maybe adapt it to the pool of oligos that you want to synthesize?
Yes, so that's a very good question. And in fact, this is what I tried to mention in my outlook, in fact. So I wanted to, so here we, to be fair, we did not look at this case. We looked at the case where we say we fixed the synthesis sequence, and now we're trying to maximize information rate for this given synthesis sequence. But in many cases, actually, it is a good idea to choose the synthesis sequence depending on the sequences that you want to synthesize. And so we do not yet have an answer to this question. But what I can tell you is, so there is the other work by Makarychev and et al., who looked basically at this problem where you have many sequences, and now you're trying to put them into batches, and each of these batches you can use like a single optimized synthesis sequence for all of these batches. And then you're synthesizing each of these batches alone. So that might be an interesting paper to look at. Yeah, to be fair, we don't have any results on this yet, but we believe at least maybe we can use some of these techniques also for that case.
Okay, thank you.
You're welcome.
Thank you. Other questions? Touraj, yeah, if you have a question.
Yeah, indeed, very refreshing and very interesting work. In fact, all along I was going to ask you a question until you put in your outlook, incorporate error correction. So as you correctly said, in reality, at least, you know, I'm not a specialist, but I've been told many times in the talks I attended that that synthesis is not a perfect operation and errors can happen. And so how would you, could you please a little bit provide some more insight? How would you incorporate error correction?
So yeah, thank you for this question. It's very good. So one way how one could incorporate error correction is the following. So the problem is if you use an error correcting code, so you have 2 options basically that one thinks of straightforwardly when you want to have error correction and efficient synthesis. So first you could encode your sequences such that you have a good synthesis time, and then you encode those sequences with the error correcting code to have error correction, but this way you are destroying maybe your good synthesis time properties. And the same thing also holds when you do it vice versa. You first encode with error correction, then with synthesis time, you actually might destroy your error correction capabilities in the second step. So one straightforward approach how to do this would be to do first, for example, encoding for efficient synthesis, and then using systematic error correcting code, which means that at least the systematic part of your sequence, it will be efficient in terms of synthesis, and the non-systematic part might or might not be efficient in terms of synthesis. I think there are probably better ways how to do this, but I'm not 100% aware of how to exactly do this.
Yeah, I was thinking that because capacity, you can define it also as a function of the probability error of the errors, right? The probability density function. And I thought that you were hinting into this type of direction. So have a definition of capacity, and then create a graph which actually includes error correction. I'm not sure if I'm clear.
I think I understand what you mean. So you can.
So capacity, you can define it also in error-prone environments, correct?
Exactly.
And so if can you backward reverse engineer and come up with some sort of graph that will actually take into account the fact that some of the codes could be actually flipped to some other A or C or G or T, and have some sort of special edges associated with that.
Yeah, that's a good point. I need to think about it for a second. So I think there are. I'm not sure if the methods rely on the on actual graphs, because in a way the graphs in the graph you have a very special structure that sort of imposes your source in a way to the channel, because you're having a cost constraint graph which where you're only allowed to use a specific cost constraint on the graph. But then if you're adding the errors, I'm not sure if one can incorporate this into the graph, but there are definitely works where, for example, where the problem is analyzed for cost constraint, sorry, not for cost, but for constraint systems that go through erroneous channels. But to be fair, I'm not 100% aware, or I'm not 100% sure how the techniques work there. So I would have to look into this, but it's definitely something interesting to look at and understand.
Especially the weights that you have in the graph can become stochastic. That's really where I'm hinting at. But maybe we can take it offline.
Okay. Thank you. Yeah.
And, we're a little bit beyond the schedule. So.
Okay. Thank you, Touraj. Other questions? I have just a small question regarding the constraints. So you talk about inserting constraints and you talk about homopolymers, but there are other constraints like a percentage of GC content that must be lower than 80 content, for example, or no pattern repetition in the synthesized strand. So can you take into consideration this constraint in your graph, or whatever the constraint, and also can you mix several constraints inside the graph and the capacity of that?
Yeah, so that's also a very nice question. So there are definitely graphs which ensure, for example, balanced sequences, as you mentioned them. So you have balanced AC and GT content. So you can I cannot draw it by head right now, but you can construct graphs that ensure sequences which are balanced. And then you can, in a way, if you want to combine two different constraints that you have. So say you have a graph for the one constraint, you have another graph for the other constraint. And then you actually, what you do exactly is you take the label product. So very similar to what we defined over here. It's just in this case, you don't have any costs because the both graphs, they don't have costs by nature, they just have labels. But if you take the label product of these two constraint graphs, you will get a new constraint graph, which consists of both constraints. And then you can use this, no problem. So that definitely works, yes.
Okay, thank you. I think it's time now. We start 10 minutes late, so we are on time. So it's time to close this session. So thank you everybody for attending this fifth workshop. So maybe we will have another workshop in some months. I don't know. We have to decide. Touraj, maybe you have some words to add, or it's fine for you.
I don't have anything to add except that thanking you, Mark, for organizing yet another great workshop with fantastic speakers on great topics. Hopefully, Andreas and Anthony, who had to leave early because he had to go to bed, can help us to reflect some of the things that they presented to us in our use case and requirements document.
Okay, so thank you everybody, and see you soon, and have a nice weekend.
Thank
Bye-bye.
you. Bye.
Thank
Bye-bye.
you, you too. Bye.